Great. Can everyone hear me okay? Great. So this is a bit of a battlement, so I'm going to sort of move over this way, so hopefully you can see me. So we're going to spend the next few minutes sort of, you're going to get to know me, but maybe I can get to know you first. So how many people are here from Cambridge? Hands up. Most of you. How many people have flown in from elsewhere? A few, yeah. I mean, that's commitment. So I've traveled up here from Stapleford, so the worst I had was a little bit of congestion on the Fen Causeway. That was fine. So no, that's really great. This is my first time presenting at the Cambridge Alumni Festival. But of course, the alumni of Cambridge University are incredible people who are sprinkled throughout the globe, and who really, and it's really great to see sort of the passion of sort of re-engagement with the institution and our sort of generation of new knowledge and our continued commitment to trying to disseminate that knowledge, but not just to our alumni, but to to the public and to the world. So, anyways, we'll get started. So I'm Mike. I'm a professor of systems genomics and population health at the Department of Public Health and Primary Care. I sit down across from Addenbrooke's on the biomedical campus, and my sort of main, we're on academic time here, so I'll just sort of stream in. I'm usually on academic time anyways. Yeah. Oh, great. So we've got, we're going to have to sort of cozy up a little bit. No worries. Is that it? No worries. That's okay. Yeah. We'll just spend a couple minutes here. There are lots of seats around that side. All good? Okay, well, welcome our new arrivals who probably don't know about or don't care about genetics, but hopefully you all care about disease. Everyone's got their coffees, great. Okay, let's get going again. I'm Mike Inouye, professor of systems genomics and population health at the Department of Public Health and Primary Care. I'm going to talk to you today about the genetics of common diseases, and that's maybe a little bit in contrast to some of recent news about potential therapies for Huntington's disease, which is incredible, amazing stuff, but it is a very rare disease, and the architecture of those diseases is fundamentally different than common diseases like heart disease, diabetes, and that sort of thing. So I'm going to tell you a little bit about the much more difficult case, actually, about what we do about diseases that somehow are allowed to become common in a population. Anyways, so I spend my time thinking about things like this. And this is something called structural complexity from Willard Johnson, who is apparently a very famous artist. I don't know much about Willard, but I really connect with this piece because, to me, this looks like what's going on inside us at the molecular level. It looks like molecular pathways, it looks like metabolism. It's very, very complicated. There are circles, cycles, routes. If you take the colors away, it could potentially be like a road map, literally like a road, like a map that you would use to drive on from London to Edinburgh, that sort of thing. And trying to disentangle this, understand what are the sort of control points, the rate limiting factors, what is sort of the basis for how things might go wrong? Where do things get sort of stuffed up? And all of that is part of my interest in genetics, because there's biochemistry here, but a lot of the constraints are about what's in DNA. So people are probably familiar with the human genome, and that the human genome sort of comprises the totality of DNA in nearly all of the cells in our bodies. And it sort of comprises a set of instructions for how you create and how you sort of manage a human being as they live their life. And these are 22 chromosomes called autosomes, plus one set of sex chromosomes, 2 X's in this case, but there is also a Y for the men. And here you see sort of in a microscope the sort of broadest view of this sort of structural complexity. How is the DNA sort of packaged and organized and sort of neatly arranged in a microscope in our cells? But in these chromosomes is sort of packaged up this DNA. And why do we care about this DNA? Well, it has a really strong influence, in many cases, on how we look, how we might, our health might, what happens to us over our lifetime in terms of our health, how we might feel. These are sort of the things that affect our DNA. And on the left here is a very famous study, a long time ago now, comparing monozygotic twins who share all of their DNA and dizygotic twins who share roughly half of their DNA. And you can see just from looking at these different sets of twins with different proportions of DNA, how similar they are or how different they are, right? The dizygotics look substantially different from the monozygotics, therefore giving you an indication about how much DNA might contribute. And we've looked at monozygotic and dizygotic twins at huge scales now. And a study now, actually 10 years ago, basically came to the conclusion that about 50% of the differences that we see in phenotypes between individuals could be attributed to differences in germline genetics. When I say germline, I mean the DNA that you're born with, not necessarily the mutations that you acquire as you live. So that's just on average, right? There are some that are much lower than that, like 10, 20%, and there are some that are much higher than that, like 80, 90%. But on average, it's about 50%, so half-half. And that's a really strong motivator for why we study DNA, because in theory, the differences in DNA can tell you up to 50% of the differences in disease risk, in body mass index, et cetera. So the human genome contains all of these sort of secrets, and that was a real sort of motivating factor for why we invested, or the community and governments around the world invested so much in sequencing the human genome. And I'm not going to sort of go through this big old timeline here, but it's really just to emphasize that this was a massive global effort that began, decades ago and continued for, over a decade, and resulted eventually in a really sort of a good, hard, solid draft in 2003. And it was something that we could start doing, real science with, I would say. We had a sort of interim draft in 2001, and we've had some more sort of finishing going on in the years since 2003. But essentially, 2003 is really when the sort of the era of like, common disease genetics and really trying to tackle these really massive, diseases of massive population burden really started. So what does the human genome look like? It's huge. It's like 3 billion or so letters. And when you put that all in books and you put them on a rack, you're basically covering an entire shelf with text of about this size. And you can go down to London, you can go to Wellcome, the Wellcome Trust, which is there on Euston Road, and you can see just how massive it is. And it's really trying to understand in this language what's going on is an immense challenge, as I'm sure everyone here can sort of appreciate. You've essentially got 4 letters, A's, C's, T's, and G's, and you've got 3 billion of them, and you're trying to understand which ones matter for what, right? In terms of our eye color, hair color, heart disease risk, et cetera, right? So how we actually deal with all of this data is the sort of principal challenge. So the community really went through a process of trying to understand, theoretically, what would the architecture of something like coronary artery disease or heart attacks look like? And we started designing our studies in ways that sort of tailored to those hypotheses. And one of the hypotheses, which really won out in the end, was called this thing called the common disease, common variant hypothesis. And it was principally sort of around the idea that the genetic signal for common diseases, so they're allowed to get that sort of common in a population, that they would have relatively small effects, and they would have relatively at this sort of gene level, they would have relatively simple architectures. So it wouldn't be like this particular letter in that particular person is what caused their disease, and then that other letter in that person. It was really that if you looked across individuals and you grouped them by cases and controls, that you could sort of arrive down on like the causal allele for, statistically at least, one that sort of causes it for all of them, right? So you don't have this massive heterogeneity in which letters cause which diseases in different people. So that meant that we could invest in coming up with things called genotyping arrays. So this is getting in a bit technological, but the idea is that you can put all of these different spots on this little slide, microscopically, and sort of run people's DNA over the slide. It'll stick to those little spots that you've said. And then each spot tells you which A, T, C, or G is at which position in the genome. And you can fix those. And you can then scale that technology really, really well and really cheaply for thousands and thousands of people, like tens of thousands, hundreds of thousands. And then you can run things called case control studies, or as we sort of call them here at least, genome-wide association studies. So we can put 500,000, a million plus dots on these arrays, run them for thousands and thousands of individuals, look at people who had disease and didn't have disease, and then we can statistically look at the differences in these alleles at different positions in the genome. We can plot it out here on the x-axis, right? Chromosomes, 1 through 22 at least, and look at the statistical signal, right, at each one of these genes. And some of them are labeled. Some of them labeled in blue, some of them labeled in red, that sort of thing. And that sort of tells you how different that particular allele, that particular gene is between people who have heart attacks and don't have heart attacks. Yeah? Clear as mud. So, but the point is that we were able to come up with essentially these maps of the phenotypic associations across the genome for many, many, many different diseases and many, many different traits. And we started running these studies at massive scales because essentially we had the technology. All we needed to do was essentially run it for lots of people, do these studies, and create databases, right? And one of the databases we created was the Genome-Wide Association Study, GWAS, catalog. And anyone here can actually go to it and access it and look at the summary statistics for these studies, which is really wonderful. This is essentially open science. And we can essentially look at, and you can't really see them here anymore, but these are the chromosomes that I showed you before, at least the 22 of them. And they are covered in all of these phenotypic associations. You can't even see the chromosomes anymore because we've done such an immense amount of mapping over the past sort of 20 years or so. And here, there's about 7,000 or so, earlier this year, 7,000 publications in journals, which have identified 780,000 plus associations between genetic variants, these A's, T's, C's, and G's, and traits, right? And there's over 100,000 full sets of summary statistics, essentially stuff like this, across different traits, different studies, sometimes the same trait, but with different sample sizes in different populations, that sort of thing. Over 100,000. So we've got all of these maps. It's open data. Anyone here, with academic researchers, citizen scientists, you can mine these data to try and identify which genes might be causative of what disease, right, or be associated with what trait. So this is a really huge advance. And this is really, and just to really draw the contrast here, this is fundamentally different than Huntington's disease, right? You don't have this one massive peak telling you that it's this, CAG repeat on, I can't quite remember, I'm not a Huntington's researcher, but this is signal that's spread right across the genome. And there are some diseases and some traits which are even more diffuse than this, right? They've got more than, like hundreds and hundreds, sometimes thousands. If it's human height, we've got, I think, 10,000 or so different independent associations across the human genome. So you get this spread. The signal gets spread across the genome. And that's really quite important for why it's allowed to become so common in a population. You diffuse the signal. Evolution can't act on one spot to get rid of that allele, but it's sort of allowed to sort of persist. Anyways. That's for the evolution people in the audience. With all of these maps, over 100,000 maps, we can look at every one of those associations and ask the question, do each one of those alleles that's associated with a disease, how big is its effect? Like, does it double our risk? Does it triple our risk? Is it something really profound, like 10 times the risk? And the answer is essentially no. We get these really, like I said, diffuse effects across the genome. And so the list of putatively clinically actionable variables, variants, that we had before the genome-wide association study era is essentially the same now with all these maps. And to some people, that was a real disappointment, right? Because they expected that each one of these new alleles that you would find, that suddenly they would just join this list and join sort of the clinical genetics services, and we would suddenly be able to utilize them in clinical practice as we have other alleles. But the fact is that a lot of them are low effect, but they're relatively common in a population, right? So how do we deal with things that frequently happen, but only sort of nudge our risk in different directions? Any sort of former statisticians in the audience or mathematical people? You aggregate that signal, and we have tons and tons of models of how we combine all of these different variables together and create things like scores, right? We do this. We do this as part of NHS Health Check. Do people do their NHS Health Check here? I haven't gotten my letter yet, so I think I should probably start a stat. I'm just guessing. But the point is that NHS Health Check and some of the risk scores that they calculate for you guys, like Q risk for cardiovascular disease, right? That's the same idea. They take a bunch of variables, they weight them up together, they sum them, they come up with a number, and that number is sort of proportional to your sort of 10-year absolute risk of having a cardiovascular event, right? And we do the same thing, just with genetic variants. So fundamentally, it's the same idea. It's stuff that we use in clinical practice. It's just a different paradigm. It's not just like one allele and suddenly you have it and then you have to, my gosh, that's really just informative. You have to sum up all of these weak effects and come up with a score that gives you a distribution in a population basically like this. And you mostly care about the whole distribution. So if you're really quantitative, you care about the whole distribution because it tells you things. But for most people, what you care about is at the tail, right? The people who are at really high risk. And for heart disease, for example, people who are at really high risk are at roughly 8% of a European population. 8% or so is at threefold increased risk of heart disease if you're sort of in this top 8%. So that's essentially the same as if you have like a familial hypercholesterolemia variant that travels in families and that sort of thing. So you have a threefold increased risk of having a heart attack. So that's sort of what this sort of means. And this is now what we call a polygenic risk score or a polygenic score. Some people might have heard that term. Have people heard that term before? Polygenic score? No, yes, sort of nods into it. Well, probably about 20% of the audience, I would say. That's not bad. That terminology, God, I gotta tell you, there were wars about this terminology like years ago. I was on the genomic side, but polygenic, what is that? But apparently people sort of hear that and they kind of comprehend it. Anyway, so that's good. So polygenic scores, what do they look like across different diseases? We take all of these variants, we sum them up. Can I actually tell you anything useful? So this is a little bit technical, but what I'm showing you here are kind of like inverted Kaplan-Meier curves, if you guys know what that is. So essentially, if you look across age here on the x-axis, and you ask, well, what proportion of people who are in the top n percentile of something, in this case a polygenic score, what proportion of those people have disease X, right? And you plot that out. We can plot that out in the sort of the top, say, 3% or so of a polygenic score and put that on the red line, right? So if you're in the top 3% of a polygenic score, on average, you're following this red line for these different diseases. If you're in the bottom, the most protected 3% of a polygenic score, you're on the green line, right? Red, blue, green. And sort of what you really want to see, and the blue is sort of the population median, what you really want to see is you want to see big differences between the red and the green, between different diseases. That tells you that you've got a risk factor that really helps you stratify cases and controls. So Some of these diseases do a little bit better than others, and actually they're all on the same scale, right? So it tells you a little bit about how common these diseases are, so it's a little bit hard. So you have to look at sort of the relative difference between between green and red here. But for some diseases, say prostate cancer for men, you get really quite differentiated curves, which I don't know about you guys, but I'm quite interested in that one. There's ischemic heart disease, breast cancer for women. I'll show you a little bit more about breast cancer and prostate cancer here in a second, and heart disease. But we can do this for over 100 diseases. You get these curves, and they look more or less like this. So potentially informative information. So that's good. We've got something that potentially could tell us something about disease risk. What are its properties? So we're looking at germline DNA here. Probably many people are familiar with genetics and germline genetics. You are sort of graduates of Cambridge, which is sort of like the home of molecular biology, and anyway, whatever. You can calculate, so it's germline genetics. You can calculate polygenic scores early in life. You can calculate it at conception, potentially, nearly at conception. And you can actually calculate it after people pass, as long as you have DNA. So people have actually been calculating polygenic scores for a lot of ancient DNA as well, trying to understand genetic risks of different diseases throughout human history. So it's really quite a unique risk factor in that regard, right? It's not like body mass index or like LDL cholesterol levels. Genotyping arrays, the ones I mentioned before, they're relatively cheap. They're 20 pounds in research studies. In real life, they're more like between 100 and 200 pounds in, say, clinical practice. But at least if you're a research study, 20 pounds is essentially, what is that, a pub dinner with a nice bottle of wine that you split with your partner, something like that. And that's a one-time cost because these scores do not change throughout your lifetime. And many PRSs, 100 or more, can be calculated from one array. Right? So it's a one-time 20-pound cost per individual. So the properties of these polygenic scores are really quite unique when it comes to risk factors. Even though they may be just risk factors, you can't do this with hardly anything else. So it could potentially have, just by virtue of the potential power of the risk factor and their properties, it could have population-level impact, I would say, if you're looking at these things. So what are the potential benefits? Well, disease risk prediction, obviously. That's what I was showing you before. There's been a lot of studies, actually, in autoimmune diseases, particularly ankylosing spondylitis. I'm not sure if anyone has experience with ANK-SPON. It's a terrible disease. Type 1 diabetes, absolutely. People probably are familiar with type 1 diabetes. Even celiac disease. So the thing about those is actually the polygenic scores are so strong that they can contribute to refining the diagnoses. They can actually inform the diagnostic pathway. In the case of ankylosing spondylitis, it's actually the single most powerful discriminatory factor that they have. It's better than MRIs. So it could potentially be used in that setting. It can tell you a little bit about disease progression and recurrence. It can tell you a bit about population screening. I'll show you a bit about that. In theory, it can prompt risk-reducing behaviors. The literature hasn't really borne that out yet, although the studies haven't really been well-powered. There is evidence that it changes clinical behaviors, like medication initiation and adherence, right? Because if you sort of think, I'm at genetically high risk, right? You actually tend to want that preventative therapy or a therapy that will slow the disease progression, right? And you'll adhere to it better. And also it can help in terms of clinical trials. And there's some studies from various sort of pharma companies looking at how they actually use polygenic scores to guide who they might actually target a drug to, right? Because you have both people at increased risk that you want to treat, and it looks like for some cases, in terms of like statins and PCSK9 inhibitors for heart disease, that if you treat people at high polygenic risk, you actually get greater risk reductions than you do otherwise. So some interesting stuff there. So those are some of the potential benefits, and I'll tell you about the risks in just a second. And that stopped working. All right. And none of that works. I have actually a yellow alien flashing up here. Does that matter? Not able to change the slides. Oh, the battery is running low. Yeah. You might want to plug in your PC. Sorry. No. Mm-hmm. Just another interlude here, actually. It's okay. I think we're restarting, are we? Okay. I actually can't remember what the next slide was. Maybe I should have looked that up. I have it on my Google Drive here. I can. You probably can't see that, but. So let's see.
Do mutations during your life change it?
The somatic mutation. It's a good question. I think that's essentially at the border of the research at the moment. So what does germline and somatic variation look like if you were to combine it into it? And there actually are some studies. It's a really good question. All right, see if that works. Oh my gosh, great. Yes, we're going to get to prostate cancer. Okay, we'll come back to that at Q&A maybe. Prostate cancer is really interesting. This is probably the most impactful study that's looking at potential clinical utility of polygenic scores that's out there. So if you have access to the New England Journal of Medicine, have a look. So this one essentially took people who, so they invited people who were at high polygenic risk of prostate cancer, so the top 10%, and they asked them if they wanted to join a screening program to see if like amongst those top 10% PGS, whether they actually had prostate cancer. And they put them through the standard of care in the UK, which is MRI and PSA screening, prostate-specific antigen. And alongside that, they looked at polygenic risk. And they asked, well, what percentage of the people who were classified as high polygenic risk were cases? That's 187, which are sort of plotted here along the top. All of them, of course, have a PGS, and this is sort of the Gleason score. It tells you the severity of the disease, red, bad. And then you've got sort of the different sort of types of standard of care here. And you sort of notice something straight away, right? There's a lot being missed by the standard of care, right? So amongst these like bona fide prostate cancer cases that polygenic score flagged, this sort of the usual screening scenario is missing them. And they're missing, the majority of them. So what this is sort of telling us is that it could have, the PGS for prostate cancer could have a pretty significant role in improving the effectiveness of prostate cancer screening. And we know there's tons of problems with prostate cancer screening, right? PSA itself, right, is both missing a lot of cases and it's, a lot of false positives. But if you combine that and an MRI, you could potentially do a lot better and detect a lot of undiagnosed cases in the community. So that's why this study got into essentially the top medical journal in the world. And I think really sort of energized a lot of people in thinking, potentially we could use these things to transform screening and identify people early on who have prostate cancer or prostate cancer risk and try and prevent it. Breast cancer as well. Probably won't go through this in a massive amount of detail, but Cambridge has been at the forefront of developing breast cancer risk prediction tools, things like Boadicea, which is a risk model, and CanRisk, which is a cancer risk prediction tool. You can go to canrisks.org. And essentially, all of those things, in sort of the process of developing those, it's become clear that polygenic score for breast cancer is actually the single most powerful individual predictor of breast cancer that we have. So for example, this is one way of looking at risk. So if you sort of look at across the distribution of a risk factor, you can essentially say, okay, well, this is a population level, right? If you just look at, just what's the frequency of the disease in the population, you get a straight line. It's a red line. And what you want when you start layering on more information is you want to see the cumulative risk distribution start stretching really high at this end and really low at that end. And that sort of tells you how good your risk factor is. And essentially, when you do that, you see that PGS is really quite powerful, but that we combine all of the risk factors together, age, sex, known pathogenic variants, BRCA1, BRCA2, et cetera, you get the most powerful risk model out there. But really, it's become clear that that you can really improve the effectiveness of your risk model by including a PGS for breast cancer. And so if you go to canrisk.org, you can actually see, and this tool is used in primary care worldwide. So on the right, there's been over 3.3 million risk assessments using CanRisk since 2019, and that's during the pandemic. So this tool is, and it's ready. It essentially has a polygenic score integrated. It's really just a matter of making that information available for individuals, and it can slot in essentially straight away into CanRisk in principle. If you really want to ask how it's going to work, you can go talk to my colleague Antonis Antoniou, who's been leading this work. So some of our work early on as well was looking at heart disease. As you probably can tell, I've been talking a lot about heart disease. And this is one of the earliest papers, probably the earliest, that was really sort of thinking about these sort of risk trajectories that I showed you before, the blue sort of red and green curves. And essentially what you can see with polygenic scores for heart disease, even years ago, nearly a decade ago now, is you could get polygenic scores that sort of get you to a clinically meaningful level of risk, 10% or so. So if you look at sort of the bottom sort of quintile, bottom 20%, and compare it to the top, you've got, what is that, 18, nearly 18 years of difference between the top and bottom 20% in a population when you actually meet this clinically meaningful level of risk. That's a huge amount of difference. And if you have sort of 18 years plus of statin usage ahead of you, your LDL levels are going to be a lot lower than they otherwise would be, and your risk of heart disease will be a lot lower as well. So that I think was really sort of meaningful to us at the time. And if you sort of look at how we reclassify risk as to whether you treat or not, you end up getting these quite substantial improvements with nearly 35% of people being correctly up-classified in terms of their risk or correctly down-classified in terms of their risk. And if you look categorically, those people that actually end up being treated or not treated, it's about 12% of people are actually correctly treated, correctly not treated, or correctly treated. So it really substantially improves the efficiency of the risk model. How do people react when they receive polygenic risk score information? So a bunch of studies have been done on this now. Generally, people receive the information as they would other risk factors, kind of like they would monogenic variants, but in a much more sort of mild way, right? Because it's sort of proportional to. It's not sort of telling you that you're going to get Huntington's disease. It's telling you that you've got like this incremental increase in risk, right? And you may want to take some sort of preventative action. So people seem to receive this information in a pretty reasonable way. But what do they actually do with that information? And I mentioned that they don't necessarily have vastly different lifestyles, and they start exercising, they transform, they lose 100 pounds or anything like that. It's not like that. But they do make some more, potentially more beneficial clinically related decisions. They decide to initiate medications more. They decide to initiate statins because that's a joint decision, right? You make that together with your physician. That's a really sort of personal decision. But if physicians are able to communicate to you that you also have genetic risk alongside your phenotypic risk, that might really, you might take that information on board even more than you would otherwise, right? And that's generally what happens. So people initiate statins more often, and they adhere to statins more, about 15% more adherence to statins over the coming years, right? And it's very difficult to motivate yourself to just keep taking that constantly, but just the effect of this information really seems to keep people staying the course essentially. And you end up seeing both reductions in LDL cholesterol, which is the bad cholesterol, which is a massive causal risk factor for heart disease, and you end up seeing reductions in cardiovascular events as well. This information is in a lot of consumer genetics products out there. So 23andMe, people have heard of 23andMe, presumably, a little bit. Maybe you have some of your own profiles done. And so this information is getting into consumers' hands as well. And people are going to their doctors and saying, what do I do about this? And societies, clinical societies, are listening to this. And they realize that this information, it's sort of, there's a fair bit of demand in the patient community for it. And there needs to be some sort of framework about how that information's received or managed in healthcare, right? So there's a realization now that this information both has potential utility, but it's already in people's hands, right? So the American Heart Association has some guidance. The European Society of Cardiology just earlier this year took it on board as well, and sort of said that polygenic scores could potentially be used as risk modifiers for people who are at sort of borderline risk, like do we treat or not treat? Well, we need something to tell us whether we do or not. PGS could be one of those, that sort of thing. So it's really starting to make its way into practice and be integrated into clinical care, which is no sort of mean feat, I can tell you. There's been a lot of policy work and a lot of scoping as to how it might be incorporated in the UK. So this is sort of some initial thinking, this was years ago now actually, about how PRS could be utilized as part of NHS health check. So amongst all the usual risk factors, cholesterol, physical activity, blood pressure, alcohol use, smoking, et cetera, it's just another risk factor and can help sort of improve the efficiency of all of these risk pathways and risk assessment, how we manage it. So this is all sort of developing, and I'll tell you a little bit more about that in a second. I wanted to cover also some of the risks of polygenic scores, because there are substantive risks, but what are they? And there's some theoretical risks that haven't been borne out, but there are also some theoretical risks that do look like they could be an issue. So there's obviously a risk of sort of false positive results, right? So this is a risk factor. Not everyone with high cholesterol is going to go on to have a heart attack, right? And it's the same sort of thing with polygenic risk, right? So you tell someone that they're at high polygenic risk, how do you sort of make sure that they understand that risk and that doesn't mean that they are definitely going to go on and get a heart attack, right? So it's not, you take it sort of outside of that sort of false positive paradigm and make sure that it's probabilistic. There's a failure to convey uncertainty a lot of times, so we don't necessarily know, what whether someone who's at, borderline polygenic risk, whether it's high or low. There's biases in terms of representation, so demographics, so ethnicities. So there's variation in terms of how powerful a polygenic score is for, say, people with African ancestry versus European ancestry. That's down to a lot of in terms of the representativeness of the data. So we have lots of European ancestry data that we're able to do our modeling on, but we don't have a lot of African or Asian ancestry data to do our modeling on. And that comes through in this. And that happens with a lot of risk factors, not just PGS. It's sort of a pernicious thing in how we do these things. So we need to develop more data. We need to generate more data so that we can have better representative models. Healthcare professionals, getting this information into healthcare professionals' hands is sort of, that's an issue, but also how do we actually train healthcare professionals to use this data? That's a huge, huge challenge, and it's not really solved. That I think will be a real sort of barrier to whether we're able to use genomics at scale for healthcare. Communication of PGS results, that's still very much an open question. So how do people receive that information? Do they need sort of like a traffic light system, high, medium, low? Do they need a speedometer? Like, how do people receive? I mean, showing you those Kaplan-Meier curves, people probably kind of like, what does a curve mean to me? People receive different information in different ways, and that sort of area has not been worked out yet, how best to do that. And there's also potentially psychosocial effects. So people who are told that they're at high polygenic risk for a really serious thing, are they anxious, right? Do they go on to develop depression around that? Those are all risks. There's currently, so that's been looked at substantially now, and there's no real evidence of that's what goes on. People tend to receive this information and sort of take it on board as they would other risk factors. So there's nothing sort of special about polygenic scores in that space. There is also a little bit more. I'll skip over the demographic stuff actually because I think I fleshed that out. There is also a dark side to human genetics, and people who are in Cambridge also probably recall that we've had some issues with this in the past. Things like genetic determinism or eugenics, right? People probably have heard the term eugenics. Has been a really horrific philosophy that's underscored some of the worst of humanity over the last sort of 100 years or so. Polygenic scores skirt sort of the edges of that, right? Because they're telling you about real things, real differences that between individuals and between groups of people. But we need to use that information in an ethical manner because it can slide into stuff like this, idiotic behavior like drinking milk because you've got this lactase tolerance allele and you're able to tolerate milk, you're sort of European ancestry. So you drink milk to emphasize your racial superiority. This actually happened. This is a real photo of men with their shirts off drinking milk in the US. And it was on the cover of the New York Times. So this information can really make people do weird things, silly things. And so that's an issue. And that we don't really have a handle on how to tackle that. How people are receiving this information and who are a little bit extreme and a little bit on the fringe. I think that's a broader sort of societal issue, even at the moment. But there's also some commercial interests that are really developing around polygenic embryo screening that are really worth thinking about. And is this a risk? Is this a potential benefit? It's really unclear at the moment. But how you actually go into embryo screening, it's such a personal thing. And it's such a potentially important thing for a population as well, because you're starting to change the germline genome of populations, right? You're starting to nudge it in directions that people might not want to go, or that societies might not want to go. Because there's not just polygenic scores for heart disease and diabetes, and you're trying to get rid of these things from a population. Maybe that's ethically defendable. But there are also polygenic scores for IQ. There are polygenic scores for height. There are polygenic scores for physical traits. And how you sort of select for those at the embryo phase has major ramifications. I mean, we're talking really thorny ethical questions. But commercial interests are really trying to push this, and we need to be careful about that because that is a very, very difficult and thorny area at the moment that I know a lot of scientists are worried about. So the point is that we've got this balancing act. We've got risks, we've got benefits. How do we put guardrails on this new technology, as we would any new technology, so that society and individuals and groups benefit from this new technology? That's the stage we're at the moment. And it's particularly important because polygenic scores in genomics are part of the NHS's new 10-year plan. Not just part of, they're on the cover. And some of the things that are being proposed or being aimed to do within this decade are really quite ambitious and have the potential, as I've hopefully shown you, to transform how we sort of predict risk, how we intervene, how we detect disease early. It can really have massive health benefits for populations, but there are these risks as well. You can probably read this quote. Polygenic scores have factored into the NHS's 10-year plan the creation of a new genomic population health service to try and actually bring genomics to the masses to improve the health of populations. All of this is really sort of laudable goals, but there are still issues that we need to work out as well. So we have this new technology. We need to be careful with it. Hopefully I've sort of convinced you that we've gone on a bit of a journey with the human genome. but that we're actually hopefully very close to having something that improves the lives of patients and the public. Thanks. I think there was actually one hand that went up. Yeah.
Given that these common diseases are probably the most, let's call it the heaviest financial burden on nation states all over the world for dealing with heart disease and so on, why would those same nation states not want to encourage embryo screening to, let's say, remove or mitigate the risk of those diseases? Isn't it a straightforward answer?
We'll put a little box around the scope of that question. That is a question that's being asked in the top sort of journals at the moment. So you're right on the edge of the research. If it's just about disease, I think you can defend that on the face of it. But when you look a little bit under the hood, a lot of these diseases are correlated in terms of their populations. And they're correlated with other diseases, and they're correlated with other traits that aren't necessarily related to disease at all. Things like, I mentioned IQ, right? A lot of diseases are associated with IQ as well. So when you start screening and removing embryos that are at high polygenic risk of disease, you actually are potentially, we call it pleiotropy. Some people probably know pleiotropy. You're actually indirectly selecting against and for other traits. And that space is very complicated indeed. And it's really hard to unpick that. And you can't go back on it. So we've got quite the puzzle. So there's a lot of people who are doing some really edgy research on polygenic embryo screening. So to be continued, but I think there's some real questions that will need to be asked of the public later on, once we know a little bit more. Yeah.
I hope people forgive me a quick advert. If you want to learn more about genetics, you can go to the Institute for Continuing Education, turn up here for half a year every other Saturday and get an undergraduate certificate in genetics, and it's loads of fun. My question was going to be kind of related to that as I recall, sickle cell anemia is kind of developed evolutionarily because it helps you be more resistant to malaria. So is there a kind of a sense related to your point that if you get rid of all sort of the obesity factors, it turns out that the other end of the plank springs up, so to speak, and you've increased some other problem?
There's no free lunch. I mean, even in genetics, there is no free lunch. Other good examples of that's a good example, APOL1 and chronic kidney disease in African Americans. Of APOL1 variant increases your risk of chronic kidney disease, but it protects you pretty substantially. Sorry? Oh, is it, sorry. Is it still on? Okay, great. So APOL1 and chronic kidney disease in African Americans is another good example of that, where these alleles increase risk of chronic kidney disease, but they're immensely protective of African sleeping sickness and trypanosomes in Africa. So you get this interesting environmental interaction. You take people out of an environment where they're exposed to all of these really awful things. They develop protective risk factors. You put them in a different environment, an obesogenic environment, and all of a sudden those things flip and they become, so it's really complicated. I want to go all the way back to the first slide that I put up about structural complexity and just really allowing that, things like that, concepts and pieces and art like that to humble you is something that I think we need more of, because these are really difficult puzzles to unpick. Yeah. Yeah.
And I know that's partly because, I mean, I've got Parkinson's and I've had it for 12 years. And I know there's no particular one gene which causes that. It is a disease which is going absolutely wild at the moment. I heard that when I got it, I had a statistic of 1 in 400 people had it. I heard 1 in 35 recently. I'm sure it wasn't. I'm sure they're from different sources and what have you. But also, I don't have the gene responsible for it, but my father had it, and it seems to be that there's getting to be, as more people get it, and they're getting it younger, there seems to be more people getting it who do have a parent with it, and so there does seem to be some epigenetic link or something going on with the environment. But that's just sort of by the way that I'm interested in. But the other thing I wanted to say to you is that you said that it doesn't seem to cause anxiety or depression, and there's an underlying assumption in what you were saying that lifestyle, if you're told that you do have an increased risk of something, then improving your lifestyle can help. And it's not like that with a lot of diseases, is it? And you've got tremendous guilt attached to, will your siblings and children get the disease if you're at increased risk? And that's something which does need taking very much into the consideration, I think, with all of this, whether anything can be done about it before you tell people of the increased risk.
Yeah. Look, neurological diseases, Parkinson's, dementia, Alzheimer's, that sort of thing, very much are very difficult to know what to do in that domain. Because if you're ApoE4 double homozygote, you're going to get dementia, very likely to get dementia by the time you're 40 years old. What do you tell those people? Because you can't stop it. There's nothing they can do. So that sort of situation is something that is very difficult to know how to improve people's lives in. And it's one of these things where you have information that tells you a lot about something, but you can't do anything about it. You can't intervene. You can't actually control the situation at all. And it can feel helpless, and it can feel, and this is family medicine as well. This is germline genetics, so it's telling you something, not a huge amount, but it is telling you something about your siblings, your parents, and your potential children. So the situation in a lot of neurological diseases with genetic risk is much more complicated than what I've shown for cardiovascular disease, where we have lots of safe and effective options for preventing it. So yeah, and there's a lot. I think each one of those instances is really going to need real engagement with your physician, your healthcare professionals, to understand what is the best thing for each patient, right? I mean, there's really no other way around it, because everyone has different perspectives on it, and it's a really complicated situation. Yeah, I think you raised your hand.
You mentioned IQ link and disease. Is this Downs linked with an increased risk of heart and other problems?
So it's not so it's not making any statement about that. So it's literally just the IQ distribution in the population and how it seems to be correlated with a lot of polygenic risk scores itself. So I'm not necessarily making any statement about Down syndrome or anything like that.
But what, I mean, I wonder if you could just elucidate that a little bit more.
In terms of the IQ?
Yeah, and which?
So so I don't study IQ. So whatever I tell you is probably total rubbish. But all I know are the correlations that exist. But IQ is also not necessarily IQ. It's not just intelligence. It's correlated with so many other psychosocial things. So it's really quite hard to say, exactly where the effects are coming from. It can be related to family income. It can be related to, all sorts of different. It can be related to your postcode. So, really trying to narrow down on where what's correlated with what is very difficult when it comes to variables like that. It's different. It's a different sort of thing than disease, right? It seems to be much more easy to pick out, real causative factors of disease from the genome than it is to pick out real causative factors of IQ from the genome. Yeah. Yeah.
I was going to ask about consent. I think that's one of the concerns I have. Clearly, there are different groups, aren't there? There are fully competent adults in a clinical trial at one end, and then perhaps you're talking about genomic screening of embryos, but also children where consent is delegated to adults, but then the impacts will be on them throughout their lives, really, and what your feeling around that was.
Again, I caveat with my replies as being like, there are people who think way more about issues of consent and like real ethicists. So really, this is just my sort of rough views as a geneticist, and it's really that consent is central to everything that we do, especially in healthcare, right? And so, if you don't consent to having your DNA profiled for disease, then that shouldn't happen, right? Or if, the whole sort of kid-parent relationship is, I won't touch that actually. I'm not quite sure how that would go, even though, I myself will have to start thinking about that very soon. I kind of think that it really should be an opt-in sort of situation of like, if you want to enter this sort of world and have, a bunch of risk factors, done and integrated into your sort of projections of risk as you move forward and what actions you might take to, maximize your health, that's something that you should consent to, right? I'm not sure, are there other sort of aspects of consent that you were sort of thinking, or?
I'm thinking particularly of children, but also the commercial kits that are available.
Yeah, well, so they can. So if you pay money for it, and obviously you're sort of consenting. But it's what sort of happens to your data, right? Once it's in the hand. Is that kind of what you're thinking with commercial, especially?
Yeah, and the implications
Yeah.
for insurance in the
Yeah.
future, life insurance in the future, for example, where they want to
Yeah.
see the polygenic risk or adjust the premium.
Yeah. There are definitely legislative guardrails that are still necessary. Life insurance, I think in this country, life insurance is not, that's not covered by some of the genetic discrimination legislation, if I'm not mistaken. Health insurance is, but not life insurance. So that still needs to be worked out, I would say. It depends on what camp you fall on. And that's also it's kind of like, how do you feel about insurance? And different societies feel differently about insurance, which is kind of interesting, especially sort of in the US. But I definitely fall in the camp of that information should be, you'd have to consent for that information to be shared with insurers of various stripes. I can't even actually see which one. Maybe let's go back over here, actually, right next to you, yeah.
Yeah. Two things. The first is I've got a daughter with genetic disease who's now 45. She wasn't diagnosed till she was 17. She is immensely grateful that she didn't know until she was 17 because there is no treatment for it. Mm-hmm. So that's just an observation. The other was that I came to a lecture at one of these things a year or so ago, and they said one of the problems of population-based screening is that the parents aren't always the parents of the child as presumed, and the ramifications from that and the legal implications are potentially absolutely enormous.
No comment. But that is a well-established fact. Actually, paternity in a population is what is it? 6, 7, 8% of individuals randomly in a population, their fathers are not necessarily their fathers by DNA. Yeah.
I wondered if it helps the NHS target scarce resource, and I was thinking of the case of women, where more women die of cardiovascular disease and actually die of breast cancer, but we target a lot of resource to the early detection of breast cancer. but maybe with something like this, you could say that there was more risk of cardiovascular and put resource to that and make some choices with scarce resources.
And that is a major motivating factor, that if you've got something like this, especially because you can get all this information for not a whole lot of funds, that it can improve the efficiency of health systems and who you would, especially for demographic groups who tend to be discriminated against and who aren't sort of prioritized. People don't think of women as getting heart disease; they think of them getting, these sorts of biases. I think I showed you the curves for men and women, right? And actually, that could be a potentially useful way. It's women who are in the top sort of 20% of polygenic risk for heart attack. They actually crossed that 10% line at roughly age, if I'm not mistaken, it was like 60 or so. Whereas women who are in the bottom quintile on the green line, they hardly even touched it at all. So it potentially could give you, I would say, a way of prioritizing much better, and hopefully addressing some of these ingrained biases. Yeah, maybe one, and then maybe we can go to the back there, actually. Yep.
Thank you. On the graphs that you were showing for disease rate over age, there appeared to be, at least from my reading, a dramatic asymmetry when you look at the top 3% versus the middle 20%, and then middle 20% versus bottom 3%. So I guess, this one.
Oh, yeah.
So I guess first off, am I reading that correctly? That there is this big asymmetry between sort of the upside and downside here? And if so, does that sort of imply that we're tuned pretty well to avoid disease even in old age? And if that's the case, does that imply that for genetic medicine, we should be optimistic about reducing the risk of the worst treated, but pessimistic about improving the outcomes of the baseline?
Not necessarily. It's a very eagle-eyed observation, so well done. But actually, it's largely statistical. So there aren't sort of there aren't non-linearities here. Actually, it's quite a linear association, if you know what I mean. This is really just because of the space here. So this is from 0 all the way up to 1, right? I haven't shown you 1. I've shown you sort of 0.4. But because this line can't go down that far, it has to asymptotically sort of hit something, right? And so it's similar to this one, right? It has to asymptote at 1. So this is really sort of just the shape of this space is why you get these sort of differences in distance between these curves. Hopefully that makes sense. Yeah, I wouldn't interpret it more than that. And there's no non-additivity, non-linearity. Yeah.
Thank you. Hopefully a very quick one. Where did the first big data sets come from for you to be able to draw all these associations from?
Ooh, that's a really great question, and I love it because it's data. So the first, well, it started small, right, as everything does. It started in a very manageable manner with things like age-related macular degeneration, which has a really massive peak at this gene called C4. So we were actually able to do that with just a few hundred cases and a few hundred controls. And that was published in 2005. So that's really what started. It started at that level. And that happened with Crohn's disease, type 1 diabetes. Like a lot of the autoimmune disease came first. But the community was really forward-looking, and they thought we should really do this systematically, especially with like a shared control set. And that was the UK. The UK is wonderful at thinking this way. Other places, not at all. I mean, the US is just sort of segmented into, it's just terrible. But the UK thought, we're going to fund a really big study called the Wellcome Trust Case Control Consortium, which was, I believe, 17,000 individuals. 2,000 cases for 7 different common diseases, heart disease, type 2 diabetes, et cetera, and a shared like 3,000 set of controls. And essentially, yes, that's 17,000. Yeah, just sit down. that has been the real door opener, the one that really kicked down the door for a lot of the genome-wide association studies and a lot of polygenic scores. So if you Google sort of WTCCC, Wellcome Trust Case Control Consortium, you'll see what that's and the transformative impact that's made on this area and on polygenic score research. So that's what I mean. I'll do a special mention though for UK Biobank as well. People are probably familiar with UK Biobank. Some people are probably part of UK Biobank. That has also been very transformational for polygenic scores as well, just by virtue of the size and it's really the depth of information, and you can do risk prediction and a lot of modeling in that data, whereas in a case control study you can't do that. So WTTC, Wellcome Trust Case Control Consortium, couldn't make that next step. You needed these really big population studies like UK Biobank to do that, and that's really taken us, the other half of the course, I guess. Yeah. Yep.
So I think one I've got a question which I think is relatively simple to answer, and then there's a whole can of worms to follow, unrelated. So if I had sorry, I forget what a kind of a polygenic array test done today, and then someone does a kind of analysis of an illness that hasn't, or a disease that hasn't really been looked at before, could you use today's data kind of retrospectively, or do you need to kind of identify the genes that you're looking at before you do the test? Does that make sense?
So like sort of your data is part of discovery, I suppose.
Yeah, or I suppose putting it another way, say the first study about age-related macular degeneration, could you go back to the samples and look at heart disease in that population, or do you need to?
So yeah, I think if you join a research study and you consent to your data being linked with, for example, your electronic health records, then you could become part of discovery efforts looking at diseases and various sort of things that haven't been looked at before. And it's just a question really of whether you want to be a part of that sort of a research environment.
Yeah.
Yeah, I mean, does that sort of answer?
Yeah,
I
so
think
there's a potential. I guess my question was more thinking about if something new comes over the horizon, how
much
can you?
Probably.
Yeah.
Yeah. I mean, it depends on various things, but generally, if you've got an array done and new scores are developed elsewhere, you could probably calculate it in your data, and maybe it could be informative for your health.
The other kind of question that it's difficult to get very specific about this, but I'm assuming that other people in other areas have looked at things like, I don't know, personality traits or kind of characteristics where it's debatable if it's a disability or an illness or not, like different types of neurodiversity and so on. And so, can you say much about how much of that is going on, and B, about the thinking around the ethics and so on?
Yes, so people, there's a lot of studies being done in sort of, I guess personality traits and psychosocial stuff. I generally don't get involved in that just because a lot of the issues are really complicated. So, yeah, I suppose it's really just a question of whether people would find that sort of information helpful. I mean, there's been polygenic scores for risk-taking behavior. There's been polygenic scores for same-sex partners, and trying to find the gay gene. There was no gay gene, but a lot of that stuff is it is being done. And the question that I generally ask is, what use is it? And how is it going to be taken by, real people, once it's published and this information becomes known? Are they going to use it for nefarious purposes, or is it going to have a really positive effect, right? And you try and weigh that up against, right, the really core principle of academics, which is empiricism, right? What does the data say? And that's where tough choices need to be made. I mean, hopefully, I'm sort of skirting around your question a little bit, but it's because I don't have an answer.